Plants are not silent beings, but they communicate with each other using chemical signals that travel through the air. These signals, known as volatile organic compounds or VOCs, can convey information about environmental threats such as drought, herbivory, or disease. However, until recently, it was not clear how plants receive and respond to these airborne messages from their neighbors. Now, a team of Japanese scientists has made a groundbreaking discovery, capturing real-time footage of plants transmitting and reacting to VOCs for the first time. Their study, published in the journal Nature Communications, reveals the intricate and dynamic nature of plant communication. The scientists, led by molecular biologist Masatsugu Toyota from Saitama University, used a novel technique to visualize plant communication. They genetically engineered Arabidopsis thaliana, a common weed from the mustard family, to produce a fluorescent protein that glows green when calcium ions are detected. Calcium ions act as messengers that trigger various responses in plant cells, such as stress, growth, or defense. By using a fluorescence microscope, the scientists could monitor the calcium signals in the plants in response to VOCs. Furthermore, the scientists also used an air pump to connect two chambers, one containing leaves and caterpillars, and the other containing intact Arabidopsis plants. The caterpillars were allowed to feed on the leaves, which induced the emission of VOCs. The air pump then transferred the VOCs to the Arabidopsis chamber, where the scientists observed the reactions of the plants. The scientists found that the Arabidopsis plants responded to the VOCs from the damaged leaves with bursts of calcium signals that spread across their leaves. These signals were not uniform, but varied in intensity, duration, and frequency, depending on the type and concentration of the VOCs. The scientists identified two VOCs, Z3-HAL and E2-HAL, that induce the strongest and most consistent calcium signals in the Arabidopsis plants. This discovery is important because it provides the first direct evidence of how plants perceive and respond to VOCs from their neighbors. It also reveals the complexity and diversity of plant communication, which is influenced by various factors such as the type, concentration, and combination of VOCs, the distance and direction of the source, and the genetic and environmental background of the receiver. The opening of new avenues for further research on plant communication, such as the role of VOCs in plant ecology, evolution, and agriculture, has been made possible due to this discovery. The conventional view of plants as passive and isolated organisms was also challenged by these findings, which showed that they are active and social beings capable of exchanging information and coordinating actions with their neighbors. In a way, it is this discovery that invites us to appreciate and respect the intelligence and sophistication of plant life, and to protect and conserve the diversity and harmony of the plant kingdom.